Let's go. All right, good morning, everyone. After you did some qualitative proteomics yesterday, we'll talk a bit more about quantification today. In the first hour and a half, I will introduce some key concepts of quantitative proteomics. What do you actually quantify? What does quantification really mean? That is something that some people are not familiar with. I briefly discuss what the basic terms are that we have in analytical chemistry. Analytical chemistry has a certain tradition of dealing with quantitative information, and sometimes we forget where that comes from. And it's kind of good to review the terms so that you know what I'll be talking about. We will then talk a bit about quantification strategies. So what type of experiment do you need to do to actually get quantitative information? What does this information look like? And I will focus then the second part of this lecture on label-free quantification. What are the concepts there? You will hear about other quantification techniques, MRM, and some of the other parts of the main institute. So let's start with talking a bit about analytical chemistry. That is fundamentally the study of separation, identification, and quantification of the chemical components of natural and artificial materials. That is so far so easy. And quantification is defined as the act of counting and measuring. And then quantitative mass spectrometry, in consequence, is the use of mass spectrometers to turn the amount of analytes into numbers. And I use the reasonably vague term amount because there are different ways to actually describe and measure what we're looking for. There are a few terms that you probably have heard that are worth mentioning and defining a bit more accurately. So the analyte, that is usually what we're looking for. We're looking for some stuff that can be proteins, peptides, metabolites. It doesn't really matter. I would like to point out, however, that no analytical technique is sort of universal. So you always find what you're looking for. And that can be as broad as all the proteins, but then you won't find the metabolites. Yeah? So an analytical technique is always tailored towards one specific class or one specific analyte. And usually you find these analytes not just by themselves, but they're usually in a so-called matrix. And this matrix is everything else that is around. And that can be reasonably complex. And the matrix can significantly impact the way the whole analysis is going. You can imagine that it is easier to determine the concentration of a drug if it's just dissolved in distilled water versus determining the concentration of this in blood. An example for that would be if you start doing proteomics from urine. And what's contained in urine? Well, you're probably looking for proteins and peptides in urine, and that would be your analytes. And all the rest, water, metabolites, urea, that is all matrix. If we want to quantify these analytes, then usually in proteomics, metabolomics, we're dealing with analytes that are already dissolved. They're in solution, and that is why by amount we usually mean something like the concentration. We usually deal with fixed volumes, yes?
So it depends on your perspective. If you're looking for the metabolites of the matrix,
then and you have different types of matrix effects. For example, there are proteins where metabolites stick to. That are adsorbed to these proteins. And that would be a matrix effect of these proteins onto the metabolites, but also vice versa. And again, it depends on what you're looking for. That is why there's no one matrix in a sample. You have to define what is matrix, what is analytes in order to figure out what also potential complications might be. When we talk about the amounts, since we're dealing with solutions, we can just pipette that stuff, and we are really interested in how much is there per defined volume. So we're dealing with molar concentrations, which would be moles per meter cubed, or mass concentrations, which would be kilograms per meter cubed. It depends a bit on what is more convenient. With metabolites, we often have molar concentrations. With proteins, it can be more convenient to just have mass concentrations. And of course, you know that you can convert these units by knowing the molecular weight of the analyte. When we determine this quantity, this concentration, then that is a measurement, and every measurement can be subject to different types of errors. And while in usual language we are not very precise at distinguishing precision and accuracy, in analytical chemistry they have very different meanings. I try to illustrate that here with this target. Every dot here is some sort of measurement that we made, and here in the bullseye, that is our true value that we want to have. Now, if you hit the same spot over and over again, but it's just not your bullseye, then you have very good precision, because you're measuring the same thing again and again, but you have poor accuracy. So precision is something related to reproducibility, and the accuracy is something related to the distance from the true value. And the two should not be mixed up. One describes the bias, and one describes the variance of your sample. This here is the opposite. You have good accuracy and poor precision. So what do you think? What is better? Is it better to have good accuracy and poor precision, or the other way around? What would you prefer?
I always prefer good precision.
Opinions on that? Good precision is good to have.
Good accuracy, you can just increase the sample size and get it.
So fundamentally, precision is good to have, but you can get out of this problem by increasing the sample size. That can reduce your error. But if you have poor accuracy, then increasing your sample size is not going to give you anything.
But you sometimes can increase your sample size to increase your accuracy.
Very true. That is ideally, you would like to have both.
Yeah. If you have like a relative, you're trying to treat it versus control. If you have good precision and there's some systematic error there, but now you know the difference and you have a very good control of your standard deviation, that to me is more important than having the right value but have a lot of scatter in the data where you may not be able to pull out the difference.
That, as I said, it depends on the situation that you have. If you just can rerun your samples and you can run 3 times as many, then that is good. You can just reduce your error and converge towards the true value. But if your measurements are all over the place, then you basically have no distinction between 2 groups, the differential analysis you were referring to. So ideally, you would have both, but it's important to keep the 2 arrows apart because they're not the same, and they will have an impact on what you can actually conclude from the data. You can imagine it something like this. What you measure is part of a distribution. Let's, for simplicity's sake, assume it's a normal distribution. And then the precision is something like the variance that you have here, and the accuracy is the bias, by what amount is that shifted from the true value. And ideally, you would have a very narrow distribution that is centered around the true value. Reality is not always as kind as we would like it to be. We also talk about random errors and systematic errors in that context, but they're basically the same concept. Now, if you're an analytical chemist and you're going after a single compound, you're setting up an assay, for example, in pharmaceutical production, and you want to determine the concentration of your compound to monitor your production, Then you sit down, develop a method. You usually have to document that. And what people then do is something like this, a so-called calibration curve. We have a reference compound. We add different concentrations of that to samples, and we measure the response. For example, our mass spectrometric detector, or it could be a photometric detector, or it could be NMR, it doesn't really matter, some sort of response. And for the majority of instruments that are popular, including mass spectrometers, you will get something like this. You will get a linear response, more or less linear response, and if you then have this detector response for an unknown sample, you can go to your calibration curve and basically extrapolate what was the concentration in your sample. And these crosses here, those are the calibration samples. Now, there are a few things that can happen. The first thing to notice is that this curve is not linear for the whole range. Usually there's something like saturation at some point going on. The second thing that is more subtle to pick up is that this curve does not always go through the zero. You would expect that if there's no concentration of zero, there shouldn't be a signal. That is not always the case. Instead, what we observe is that there is some noise. And as soon as your signal dips into that noise, it becomes hard to really see whether something is there or not. That is what we call the level of detection, or LOD. Here you can say there is something, but you cannot yet accurately quantify it with a certain confidence. That happens at the level of quantification, LOQ. Here you can quantify with a certain accuracy. Typically that is higher than the level of detection, of course. Then you have a range where you can quantify more or less linearly, and eventually you get into this saturation, and that is what we call the LOL, the limit of linearity. And the difference between LOQ and LOL, that is the linear range or dynamic range of our measurement. Okay? Everyone familiar with those terms? Okay. I like to include them because people often talk about dynamic range. I've talked about dynamic range as well, and we don't really know exactly what it is. And that makes it hard to understand what the problems are. Let's talk a bit about this limit of detection. It's defined as the lowest analyte concentration that can be distinguished from the absence of the analyte, so from a blank, with a certain confidence, generally 99% confidence. And that is why you cannot talk about quantification. You're just talking about absence. You're just saying this is below a certain threshold, but you cannot say how much it is exactly. If you want to quantify with a certain confidence, you're making a statement that it is basically in that interval, and that you cannot say at the level of detection. You need to go usually a lot higher. And that has to do with also the distribution of these measurements that you get in these blanks and in these samples that are close to the limit of detection. There's an analogy that I'd like to show. Suppose you're at an airport with lots of noise from jets taking off. If the person next to you speaks softly, you will probably not hear them. Their voice is less than the level of detection. You might see that they're there, but that's cheating. You still haven't heard it. If they speak louder, then you may hear them, but it's not possible to be certain what they are saying. And only if the noise level then drops, can you confidently state either yes, they're speaking, and you can confidently discern what they are saying. And that is this idea of level of detection and level of quantification. You can detect that they're saying something, but you cannot figure it out. And if you are far enough from this noise level, from this background noise, then you can really figure out what is going on, and then you can reliably quantify. So how do we do quantitative mass spectrometry? I would like to give you some more detail on what is really going on in a mass spectrometer for that. Because in order to determine the amount of ions, we have quite a few steps that we need to go through. We have an ion source. So in order to determine the amount, everything that is in our sample, in our volume, needs to be ionized. That's not going to happen. So we lose some of that quantity already in the ionization. That's the ionization efficiency. Not everything that goes into this spray will actually end up being ionized. Now this spray is also not entering the mass spec 100%. Only a small fraction of the spray is actually getting sucked into the vacuum. So you're losing lots of material there. Then you see we have complex ion optics. Transfers, transfer quadrupoles, octupoles, you have the center section, you have the mass filters. So each of those comes with loss of material. Now in an ideal world, the amount that you lose or the percentage that you lose at each of these steps is going to be constant. But it is there, and it means that you need to figure out how to deal with it. You can also see that when we think about this process, we basically inject these ions here. We transfer them into a seed trap, for example, to hold them, and from the seed trap into a collision cell, from a collision cell back into the trap, from the trap into the ion trap, orbit trap, to measure them. Each of these transfers comes with some loss, and you can also see that there's some capacity to that. The seed trap has only limited capacity. You cannot pump an arbitrary number of ions into it. And that is one of the reasons why we see these saturation effects. So along this whole process, we are losing some of the stuff, hopefully in a consistent manner between samples. And we cannot pump arbitrary amounts of ions in there. And that gives us problems like saturation. And that is also where the matrix comes in again. Because if you haven't selected if you haven't selected one type of ions, then all the other ions contribute to the overall capacity. And they can lead to saturation effects, even at lower concentrations. Okay. When we're doing quantitative LCMS, and then we have one other thing to deal with. We have to deal with the chromatography. We are usually injecting the fixed volume of the sample with a sample loop. Everything that's in the loop goes onto the column. So ideally, everything that enters the column also leaves the column. That's what we would like to see. We know that is not always true. Some stuff just sticks to the column, never comes out, or stuff comes out at an inconvenient time, 3 measurements later. But for simplicity's sake, let's just assume everything that goes in goes out again. Then this analyte spreads over elution time. And the concentration at the end of the column depends on the retention time. That's called peak broadening. You know that at later retention times, the peaks tend to be broader than they are at the beginning of the retention time. And there's a simple model behind that that I can explain later on if you're interested. There's this rather old model of theoretical plates that models or describes this peak broadening. Assuming that everything that goes in also comes out, we can just say if we want to know what the concentration originally was, we just need to determine whatever elutes of this analyte over time. So we would expect that the integral of these intensities is somehow proportional to the integral of the concentrations if we have a linear detector response. So if at any time the detector signal is just some response factor times the concentration. That's straightforward. So ultimately, by integrating the detector signal, you can get the integral of the concentration. And that is what we are after, because we are after the concentration of everything that entered the column. Now, if we assume that these elution profiles are roughly Gaussians, they're not really, but let's not make it too complicated. Then we can model that as these concentrations as a product of a Gaussian and the concentration that really went in. what are the parameters of the Gaussian? Well, that's the time when of the apex. When does it elute? It is something like the width of the peak, and of course the time when you measure it. So we can plug that in, and it basically tells us that, well, since we know that what the integral of this Gaussian is, that's 1, we basically get something like this integral of our detector signal is our sensitivity times the total concentration that went into the column. That seems trivial, but that is actually why quantification with mass spectrometry works. Now, what can we actually quantify? When we're looking at proteomics, we often see that we can identify more proteins than we can quantify. So why is that? Because we can often get decent fragment spectra of peptides where we can no longer quantify them because their concentration is below the level of quantification. So when you think of the concentration range that we see, so you have concentration here and you have the number of proteins that you observe, then you have plenty of proteins that have high concentration and they're above the level of quantification, so you can easily quantify them and identify them. But then you have a bunch that are above what we would call the level of identification. They are below the level of quantification, but we can still figure out what it is. So we know it's in there, but we just don't know how much of it is there. And there's plenty of stuff that is below the level of detection and the level of identification. That is what we don't see. That's basically everything of this iceberg that is below the waterline. And our measurement will remain blind to that. With metabolomics, things are actually a bit different. We will talk about that this afternoon. Here it is often much harder to identify metabolites. So while fundamentally this looks the same, identification is a much bigger issue and we have much fewer identified metabolites. So in proteomics, we usually identify more than we can quantify. In metabolomics, it's just the other way around. The details of that we will discuss this afternoon and tomorrow. Now, how are these quantification experiments done? When people started doing quantitative proteomics, we didn't have this resolution. We had less reliable chromatography. Things were much worse than they are today. And people tried every trick to really reduce the error in these measurements in order to get decent quantification. And a neat trick to do that is labeling. What does labeling mean? You know it from the lab. You can label with fluorescent dyes, for example. That is something that is often done, or you can label with some tags that you can attach to them. Since we're dealing with mass spectrometers here, we have only one thing that we can use for tagging, and that is really mass. So by labeling, we usually mean labeling with heavier isotopes, stable isotopes. So we integrate additional heavy atoms like deuterium or heavy carbon, nitrogen or oxygen, and they have different masses, and we use this mass difference to keep two samples apart, or more than two, multiple samples apart. What you basically do is you introduce a label in one sample and you don't in the other. You mix them, you measure them in parallel, and then you do some sort of comparison. The advantages are pretty obvious. Both samples are treated absolutely identical, so we reduce the error by having the same error applied to each step. And that gives you a lower, well, what? Accuracy or precision? Yeah, it improves the precision of this measurement. Yeah? And that is why people like that for these differential settings in particular. The disadvantages are these labels are often expensive. They're somewhat difficult to apply in some cases. Might be unreliable to introduce because you have to introduce them quantitatively. Because if the labeling reaction only works in like 50% of the time, then that might be a problem. so there's some issues there. Also, labeling is not always possible. If you do in vivo labeling, then that might be possible in mice, but try to do it in patients. It's just not done. So how does that work? Fundamentally, we have two different types of labelings that are common. One is chemical labeling. We extract all the peptides and we do something to them chemically after extraction. That means usually we attach something to either specific side chains or the N-terminus, C-terminus. And the advantage is that it does not involve a perturbation of the in vivo system. You do it after extraction. That is why it's generally applicable also to clinical samples. One of the reasons why iTrack, TMT are so popular with clinical proteomics. The labeling does occur rather late during sample preparation, and that is why the labeling is not pulled through early sample preparation steps. So if you have variants introduced there, you will not see that. Well, you will see it, but you will not correct that in the labeling. The other option is metabolic labeling. The name already implies that you make use of the in vivo system to introduce the label. Prominent examples are Silac that you might have heard of. You introduce these stable isotopes by feeding the organism with some labeled metabolite like amino acids. And the full incorporation, of course, can take a while. That's often done with cell culture. You just grow that on the proper medium, and then you introduce these labeled amino acids. And since the labeling occurs very early on in the study, you have a pretty high reproducibility because it's pulled through all these steps in the same manner. If you don't want to do labeling, then we usually talk about label-free quantification. That's if you want the most natural way of quantifying, because you don't do anything to your sample. You just take it as it comes. So no labeling is required, which makes it cheap, and that makes it popular for clinical applications as well. If you have 1,000 samples, then the cost for labeling can also be prohibitively high. The data is acquired on different samples, in different measurements that might give you trouble, and also the manual analysis is more difficult. If you look at Silac or iTrack, you can often do the quantification just by looking at one spectrum. With label-free quantification, you need more advanced methods to actually do that. This figure here, took from a review, gives you some idea of what that really looks like. In metabolic labeling, you introduce these two labels, heavy or light, for simplicity's sake, early on already, and that can be in cell culture, but it can also be in small animals. And then you mix it already before you do the fractionation. And that means both samples are treated exactly the same way downstream from purification, fractionation to protein extraction, digest to the mass spectrum measurement. It also means that you need half the instrument time, because one run actually gives you 2 samples. Sounds like a great deal, but you also pay for that. Can you imagine what the price is that you pay for that?
Yeah, half the sensitivity.
Yeah. You have half the sensitivity, and it also you lose dynamic range because you basically have 2 samples in there at the same time, which means that from each of these samples, you have only half the amount. And that means it becomes easier to drop below that level of quantification or detection. So again, there's no free lunch. With label-free, you would do nothing to these samples except process them independently, measure the data independently, and then merge them only in the data analysis. And then there's chemical labeling in between, where you do purification, fractionation, protein digestion, and then on the level of peptides, you suddenly introduce this label, and then you process it in parallel. So for this course, we will focus on label-free quantification, but I'd be happy to discuss other quantification strategies and also how you implement that with OpenMS independently. We just couldn't squeeze more into this course, and I hope you have some understanding for that. When we look at the data that we get out of label-free quantification, then we need to go two-dimensional. Yesterday, what we discussed were just spectra of peptides. Today, we're going to be talking about spectra of peptides. But the second dimension that we will add is retention time. Remember what I showed you, we need to integrate over retention time in order to quantify properly. So you can imagine the data as consisting of mass spectra, MS1 spectra in this case, but there is some temporal sequence to them. So if this here is elution time, then you see there's there are peaks coming up, then they're going down again. That's basically a peptide eluting, and here's another peptide at a different mass that is eluting. And you can represent that in different ways, either as individual spectra, just not very nice to look at, but you've all seen in top view the data, what it looks like. This here is a color-coded intensity map where you see there are things, whatever they are here. And you can also look at this 3D view where you see stuff coming up, then going down, and you also see the isotope profiles that are attached to that. That's the data we will be talking, and I would like to straighten out a few terms that we will be using throughout the course. The whole thing, this whole data set, where we have retention time and M over Z as two dimensions, and intensity as the third, that is what we call a map. That's one run. And in this map, we have lots of signal, and we call the signal that is caused by one analyte in one charge state. We call that a feature. Yeah, so here you see there is an isotope profile. You have, in this case, it's probably about 100 different peaks in these mass spectra. Which of these dimensions is M over Z and which is retention time? Well, you see the peaks have a Gaussian profile along this dimension. So here, going from left to right, that's retention time. And this is M over Z, and you can recognize that because here you have the isotope profiles that you've seen yesterday. Yeah? So basically, we have 100 peaks or so that come from the monoisotopic mass, from the +1 and the +2. And they have both more or less Gaussian elution profiles. And according to what I told you earlier, in order to quantify the thing, I need to figure out that all these peaks came from the same analyte. I need to integrate them. And basically, the volume of this integral, that is going to be proportional to what I want to quantify.
So you always do all isotope.
I'll get back to that. But yes. That's what we call a feature. And the process of really figuring out that these things go together, that is what we call feature finding. And that is what I will show in a minute.
What's nice here is you can also see there's some endogenous contaminant that's just continually eluting right there. Yeah.
Shit happens.
Oh yeah. The braided well, it's big enough you can use it as your calibration.
Yeah, you can. I mean, there are many purposes, but for most purposes, it's just a nuisance.
Yeah.
Also, for algorithms. It would be nice if it weren't there, but there it is. We just need to deal with it. Yes.
Feature isn't always a metabolite.
No, feature is an analyte. Yeah, so it can be a peptide, it can be a metabolite. Right now we will be talking about peptides. This afternoon we will be talking about metabolites.
Can we part of the metabolite,
Yes, if it can be a broken up metabolite if this degradation occurs during chromatography. It has to be present already. It can't happen in a mass spectrometer. Because, yeah, well, we usually see tens of thousands. And you would be surprised. The problem really is that mass spectrometry is really, really sensitive. So if you take distilled water, even if you distill it 3-4 more times, you will see thousands of things that are in there. So getting rid of or measuring blanks is actually something that is good. It tells you, it gives you some idea of that you should not over-interpret if you see a small blip coming off that. doesn't mean that it is in your sample. It just means that it's in your mass spectrometer, and there are different ways for that to get there. Okay, so feature finding is a process of figuring out what comes from one analyte, and I should say that it's not exactly correct. There can be one analyte that is spread over several features. Can you imagine why that is? Adducts in metabolites, so proteomics it's less frequent, but also different charge states. You can see the same peptide with a +2 and a +3 charge, and you can easily imagine, well, you have one feature here and one feature there. They should be co-eluting, because they're basically the same thing, but they have different masses because and in one case there's a sodium ion sticking to it, in another case there's an ammonium ion sticking to it. And if you want to figure out the concentration of your analyte, then you of course need to sum them up, and that is already another problem. Okay, let's look at the data a bit more. Data can be messy, and fundamentally you would like to figure out that these peaks here all come from the same analyte. You want to quantify that. That's what we're talking about today. That can be absolute or can be relative. So you could come up with a statement like this is 50 nanomole per microliter, or you could come up with a statement compared to the other map, it is 3 times overexpressed. And that depends on your experimental setting, depends on what you're really wondering what you should be doing. Since label-free quantification is really straightforward experimentally, you would just have 100 samples, you measure them independently. The trouble is now in the data analysis. It's not in measuring that stuff. Although there are issues there as well that we can talk about later on. For example, measuring 1,000 samples on the same column is just not going to happen. But for simplicity's sake, let's assume we got 3 samples. We measure them independently. We know there are peptides in there. We did a bottom-up experiment, shotgun proteomics. We have something that's gray, or actually on my screen it's blue, and green and red. And if we do feature detection or feature finding, then what we would like to do is take these millions of peaks and reduce them to features. And I indicate them as dots in these maps. So we have retention time, we have m over z, and every dot here would correspond to one particular peptide. Now what can happen? Chromatography is the less stable part of our measurement. It is a lot easier for peaks to shift in retention time than it is for peaks to shift in mass. That has to do with a higher accuracy that we have in the mass spectrometer. So we usually need to align these maps. And by align, I mean we shift the retention time. It can be in a linear or nonlinear fashion. There are some discussions that we need to go into later on. And we do that in a way that features that are the same thing can be overlaid, that they end up in the same spot. Because then we can link them across these maps. Now we have these maps sort of basically stacked on top of each other, and we can say, well, what is this guy? That is what we did yesterday. We figure out what the protein or peptide is, and we figure out that the relative concentration in these maps are something like 1 to 1 to 1/2. And that is everything we need to do quantitative proteomics. Seems straightforward enough. When you think of this process as a computer scientist, then it is fundamentally a massive data reduction step. I mentioned that yesterday when I was talking about pipelines. A terabyte goes in and a spreadsheet comes out. You start with samples, you do HPLC-MS, and you get raw data. That's basically just the spectra that you recorded for each of your samples. We often do signal processing to do proper peak picking. We do some feature finding here, and we end up with these maps that contain only features, these dots that I showed you. Once we do this differential analysis, we can also ask things like, what are the features that are really differential between, say, two groups, or that are not in the blanks and still differential between the two groups, or that are mutually differential between any pair of groups if you have a more complex design. Once you have that, you usually end up with just a list of differential proteins. And that can be reasonably short. That's usually literally just a spreadsheet with a couple dozen or a couple hundred proteins. So the data size often goes down drastically. These are just numbers from one experiment that we did a long time ago, where we started with 10 gigabytes and we actually ended with 1 kilobyte of differential proteins. That's sort of the order of magnitude that you would expect. So we're talking about 7 orders of magnitude of data reductions through the processing of this pipeline.
It seems like the alignment of features is such a like
We will go into that. But the reason why we need to do alignment is really the instability of the chromatography. But the way we can correct for that is really through the mass, because the mass is more reproducible than the chromatography. Well, they need to be, are you talking about the wet lab preparation or the data processing?
They can just realign any samples in the line, or do we
Yeah. So, yeah, what do you think? Can you align arbitrary? Yeah, we'll talk about normalization later on.
LC gradient has to be the same from one.
Yeah. So, obviously, these samples should be comparable in order for them to be compared. That's where it comes from. If you try to align a yeast proteome with one from Arabidopsis, it's just not going to work. The underlying assumption of doing quantitative proteomics is that the shifts in the proteomes are usually subtle. The proteome never really changes drastically. If we're dealing with the same, it's a common misconception that the proteome changes a lot. When you measure the same organism, might be biological replicates, might be different tissues, then these proteomes are not vastly different. Why is that?
Biological systems trying to stay in homeostasis.
Yeah. We're dealing with homeostasis. So, for no. There's more variance, yeah, but you should not sort of try and compare a urinary metabolome with a blood metabolome. Yeah, these are different things. You can try to do it, but it's not going to work. Yeah. So it needs to be comparable, and we will actually use that later on in normalization, because there are some issues there as well. Yes.
So, in terms of feature finding, well, let's say you have 3 biological replicates in 2 conditions, so 6 samples each. How many of the samples do you need to find a single feature in of one condition in order to say that that's a real thing?
Well, the feature you find in a single run. Let me explain how we find the features, then that will follow automatically. I hope I understood your question correctly, but you need just one run to find the features. Okay, let's talk a bit about feature finding. So the key idea is really, when you look at these maps, you know where the features are. Yeah, I mean, we are good at finding patterns. We look at them and we know where they are, so can't be that hard for an algorithm. The idea would really be, we try to find all the peaks that belong to one peptide, and we do that by looking at regions that are sort of suspicious of containing signal And what are regions that are suspicious of containing signal? Well, that's where peaks are high. That's a good starting point. So you would argue, well, in this region, this here is the highest peak, so let's just see what's around this peak and figure out whether that can be sort of put together into a feature. What makes a good feature? When you look at this map, you see suddenly it's not that clear-cut anymore. And note that these small excerpts are usually around a 1/1,000,000th of a whole map. This is just a very small excerpt. Obviously, we have some signal here, so this is where we would start to look. But you could argue, well, there's probably something here, and there might be something there. The idea of most of these feature finding algorithms is to start with the stuff that is easy and then work your way down until you hit the noise, and eventually you need to stop, and there are different criteria for doing that. How would we describe that? Well, if we want to quantify it, and that's what feature detection is for, we need to reduce it to all the signal intensity of all the analytes that really come from the same, or the signal that comes from the same analyte. We would like to replace that with a model, mathematical model, that describes all the signal that comes from one peptide. And this model then can give us a few parameters. We want to know where it is, so the position m/z and retention time. We want to know the intensity, that is basically the integral over the whole thing, or the volume of this model. You want to get some idea of the quality. How well does it really look like a feature? And there are other things like the charge that you can easily determine from the features as well. So many feature finding algorithms start by assuming that we have some knowledge about how this pattern comes about. When you think about this two-dimensional model, then fundamentally it's just a convolution of two models. We have the isotope pattern that describes what the feature looks like in m/z. And you see this here is now really low resolution data, but it sort of illustrates the idea a bit better than if you have just these distinct peaks. And in the retention time dimension, it's probably going to be a chromatographic peak, a Gaussian. And if you multiply the two, you get something like this, a two-dimensional thingy feature model that you try to find. So it's basically a pattern recognition problem. How can we mathematically describe these two shapes? In chromatography, we would like things to be Gaussian shaped, and then the parameters are really straightforward. We have a width, we have a height, we have a position. Those are the three parameters. Are chromatographic peaks Gaussians? So what's different in chromatography usually? Where do they differ? Yeah, they're often not symmetric, so there's tailing or heading in these peaks. They're not symmetric, and the processes are reasonably well understood. There are many papers out there that give different shapes, exponentially modified Gaussians, for example, that have some asymmetry parameter. There are literally, in the literature, there are hundreds of models for chromatographic peaks that you can just pick. But they will all look reasonably similar to this Gaussian. They are not vastly different from it. Just a bit more asymmetric, a bit different behavior in the tails. In mass spectrometry, we talked about that yesterday. We can just model the intensities by binomial distribution. And the width of these peaks, if we have low resolution, that can be described again by a Gaussian. So we can consider these independent because they're independent separation processes. LC runs totally independent of MS. You do one first and then the other. So we can just assume if we have those two models, we can throw them together, we multiply them, and that gives us a two-dimensional model. One problem we have here is that if we don't know how many carbons, how many nitrogens we have in a peptide, we actually can't compute this binomial distribution. And this is where people started looking at, well, how different are peptides? And people came up with this idea of a so-called average gene. That is an average protein You can just go into a database. It's a nice exercise. You go into SwissProt. You just digest all the tryptic peptides, and you determine their composition. And it turns out that the 20 amino acids, different as they are, if you have enough peptides, they're not that much different. Basically, an average amino acid has a composition of C4.94, H 7.76, and so on. That does not mean that they're like fractional carbons, but it means that if you have a certain mass, if you have a mass of, say, 1,000 Dalton that you see for the feature, you can determine how many carbons should be in an amino acid of that mass. And from that, you can actually compute the binomial distribution that would fit at that particular mass. And that is why this averaging model is used or is so popular. And that's basically under this distribution that I showed you yesterday. If you assume that you have a peptide of 1,000 Dalton, you go back to the average gene mass, you compute how many amino acids should be there, how many carbons should be in there, and you compute this binomial distribution. And that tells you in this part of the map, if I'm looking at 1,000 Dalton, I should be looking for a pattern where the monoisotopic is the highest peak. And if I move up in this map in regions that have higher mass or higher charge states, then I should be looking for things that look more like a Gaussian distribution. And that is something that is a difference between proteomics and metabolomics. We'll see this afternoon that with metabolites, we don't have an average gene model. And that makes it a bit harder to find these features. So once we know that, we can also factor in the instrument resolution. If you assume a Gaussian shape for each of the peaks, then you can model different instrument resolutions just as different widths around these peaks. This is usually required for low-resolution instruments. For modern high-resolution instrument, this plays a much smaller role because you usually have baseline separated peaks in the spectrum. One algorithm for feature finding that's still in OpenMS and that we've published a long time ago looks roughly like this. It assumes that we look for features of different charge states. You would expect that the peptides have charge states of +2 to, say, +4 or +5. And that is one of the parameters of the algorithm. You can, if you expect your peptides to be have a higher charge, say because you introduced a modification or whatever, you can change that. We basically search for each of these features independently, or the charge states independently for a multitude of reasons. And what we do is we start with a seed list, and the seed lists are just peaks that have a high enough intensity. And around these seeds, we then look at the other peaks. We extend the region. That means we go up and down in retention time and M/Z and collect peaks that have sufficiently high intensities. Then we compute what model should be expected for a charge state of 2, and that's why we do that independently. If you know you're looking at M/Z 1000, you look at a charge state of 2, then you need to assume that the model has actually mass 2000. Yeah? So you compute the binomial distribution for that and try to fit that to all the peaks that you have collected. Once we have fitted the model to the region, we clip the region. So we remove everything that is inconsistent with the model and keep only those peaks that are consistent with the model. The rest we discard. And then we decide whether we're going to keep that guy or not, depending on the quality of the fit. And if we keep it, we remove those peaks from the data set and redo with the next seed. It's a straightforward process. What that also gives us are so-called mass traces. This here now is, again, each of these crosses is a peak. We have M/Z and retention time here. And what we usually see is that we have these isotope profiles that are eluding. Why are these triangles? Why aren't they like squarish? Why? This here is a monoisotopic, +1, +2. Why are there? Yeah. If this is what it looks like, then basically these +1 and +2, they drop below the limit of detection sooner. So you basically see shorter mass traces. So once you have identified the highest peak, the monoisotopic in this case, you know that you don't need to look further than a lower and upper boundary that's defined by these high intensity mass traces. The others need to be shorter than that. And that narrows down your search range. And these mass traces are basically series of peaks in adjacent spectra that have the same mass to charge. And this is where the mass accuracy of your instrument comes in. And that is why you need to know that for the feature finding process. If you have no idea how accurate the mass is being determined between adjacent spectra, you cannot really identify these peaks as the same mass between spectra. One thing that can happen is that, or will happen, is that these features overlap, and they can overlap in many different ways. Mass traces can contain more than one chromatographic peak. Mass traces can be interleaved in the M/Z dimension, and you can also have co-eluting features that share mass traces if they're just offset by integral number of protons. Resolving these conflicts is what we call feature deconvolution, and that is done by statistical testing. You can just ask the question, what is the most likely explanation for all this signal that I have seen? I give you some rough idea of what these problems might look like. These are interleaved features. You have one feature here, these dotted lines, and a second feature, these dashed lines, is sort of lying in between. That is actually the easiest part if you have sufficient mass resolution. If these mass traces can be distinguished, then that is easy to disentangle. This was a problem for low resolution instruments. For high resolution instruments, this problem has basically gone away. They're independent enough if the mass resolution is there. This here happens a lot if two peptides are isobaric and have only and have, for example, a cis-proline versus a trans-proline. Then you get two resolved features for this peptide that are overlapping because they're chemically still very similar. So when you look at the mass trace at the retention time, you would see something like this. And what we need to do, we basically need to cut it at this position and say that's one feature, that's the other feature. You should keep them separate. What can also happen is that they co-elute either by accident or because they have a modification that is chemically still similar. Can also be some labeling. But the higher mass traces of one coincide with the monoisotopic of the next one. And that is where the model fitting helps, because the model would then fit also the other mass traces and would reduce this interference effect. All right, how do we do the modeling? As I said, we do some statistical testing. We try different chart states. These chart states have different models, and we just see which of these models would fit best to this distribution. In this case here, it would be straightforward. We would try to fit +1, and that would fit nicely. +2 would not fit because there would be peaks missing. +2 would have peaks here in between. And +3, the shape just doesn't match at all. So that is kind of straightforward. You match that. There are different ways to do that and determine the parameters. Then you can also estimate the quality of fit. That is easily done by just correlation measure. You correlate for each data point what the model would say at the position of this data point. So this is similar to, this is a simple dot product again that we put in there. We normalize that, and it tells us how well does the data fit to the model. If that is above a certain threshold, we accept it. Otherwise, we reject it. Feature resolution works most of the time reasonably well, but it doesn't have to, because there are some issues where things look funny. So what is going on here? So first of all, this looks like a very clean feature. There's not a lot of noise in there. You have nice elution profiles. They're all co-eluting. The only thing that is funny is this second guy here, because from the distribution, you would expect that the +1 peak is higher. Yeah, this guy should be higher than this guy, but it's the other way around. What is going on? There are multiple explanations for something like this.
You could have sulfur.
Yeah. Remember what I told about sulfur yesterday, that you have a mass difference of +2? So this is a typical case that we have for sulfur, which is why sulfur peptides are tested independently in this algorithm. But it could also be that this here is a specific labeling that has a mass difference of 2 Dalton. And that's actually where the spectrum comes from. So this was a 2 Dalton shift introduced through a labeling, and then you see stuff like that as well. You can also have oxidation that leads to similar shifts. So it's good to know that, and it's good to be aware that these things can happen. Yeah. No, the mathematical model is going to be the same, but the parameters are going to be slightly different. And the parameters that play a role here are what do we expect in terms of chromatographic peak width, and what do we expect in terms of mass spectrometric resolution. These are basically the key parameters. Okay, problems here. Low resolution instrument might not give good isotope patterns. People still use them. You just need to be aware of that. We see that less and less as a problem. Peptides can overlap, in particular in complex samples. We easily see over 100, 150,000 features in one individual sample, and then you can imagine that there's constant overlap going on. Often low-intensity features are hard to distinguish from the noise peaks, and then you need to do some thresholding and need to have some idea of how deep you want to go. And isotope labels that you introduce can also skew this distribution and make it hard to pick these things up. So while we're talking about label-free, this applies to all types of data, and people have applied it to labeled data as well, and are then often disappointed. But if you have a labeling in there, no matter how you introduce it, for example, aqua peptides, isotope labeled peptides, that will break the model. And if you don't correct for that, then you will not find these things. Then we do the alignment. So feature-based alignment is what we tend to prefer because we're doing this data reduction. It makes no sense to align the spectra with all the peaks. That is nonsensical. Instead, you want to go to the features and align these features. Retention time can shift between experiments. There are a number of reasons for that. It's much harder to control that. You also have issues with the pumps. You have issues with temperature. You have issues with the column that only supports a certain number of injections. If you change columns in between, you tend to have batch effects. So there are many things that can go wrong. We need to do this, but there are certain limits to this type of approach. If your chromatography is not stable, you should not try to fix that using alignment techniques. You should go to the person generating the data and tell them to get their act together. I know that's easier said than done, but there are limits to what these methods can do. And if you have unreliable chromatography, then the retention time shifts are the least of your problem. Then also the rest is unreliable. The quantification is just as unreliable. So if you see massive issues, if the alignment doesn't work, then you should also do a quality control on your data and potentially rerun some of these samples. Just as a disclaimer, of course we try to correct for these issues, but there are limits to what we can do. Ultimately, this feature-based alignment is a reasonably straightforward thing. We start with these raw maps that contains all the peaks. We do feature finding, and that goes from a couple thousand, a couple hundred, or in larger data sets, couple 100 million peak down to a couple hundred or couple thousand features. And that makes it a lot easier. You can imagine figuring out how to correct for retention time between here and there is tough. But if you go to a much sparser data set down here, then this becomes a lot easier, and also you make less mistakes in assigning what's what. So ideally, in an ideal world, we could get away with a linear alignment, where we just see shifts. That can be done reasonably quick, and I'm going to introduce one algorithm. We have actually multiple algorithms in there. What you should keep in mind is, however, that you have often not just 2 maps, but you have 1,000 maps. And then it's not totally evident how you do it. So one way to do that is do some star type of alignment. You pick one map, and you map everything onto this map. But there are also other tricks. You can do some tree-like alignment, where you look at pairs of maps that are most similar, and then sort of build this up incrementally. For simplicity's sake, let's start with the simple type of alignment, where we are looking at 2 maps only. If we have 3 samples, we would probably say we compute 3 transformations, linear transformations, that take the features that we have in this map, transfer it to another map, and we try to compute one transformation for each map that maps them all onto one consensus map. And this consensus map, we can then identify which peaks belong together. Note that this alignment does not yet tell us that this peak here and this peak should belong together. It just computes a global alignment that brings them close enough that in the second step, in the linking step, we can actually identify them more easily. So we start with the simple pairwise alignment. Here we have the problem of one map with features and the second map with features, and we're asking the question, can we find an affine transformation, a linear transformation, that minimizes the distance between corresponding spots in these maps? So we're looking for something like this, where we shift all these crosses such that they come to lie on top of these circles. A very simple, straightforward algorithm for doing that is called post-clustering. Notice we don't know which corresponds to which other dot. Yeah, we have not identified them. All we have is the position. And post-clustering, in a way, tries to find parameters for this linear transformation. If we're looking at retention time only, we can do that independently. If we're looking at retention time only, we're looking for a global shift, B, and we're looking for some slope, A, that determines of how we do the transformation. And what you can basically do is you can test all pairs of these dots in a certain region, and incorrect matches will cluster all over, but the correct matches will sort of pile up at one position. It's a very efficient algorithm that can be run in under a second, and that gives you a good estimate of that. I can explain the details later on. For now, I leave it at that. But people have claimed a lot that you're still left with some residual error. So you should do nonlinear alignment. And they tend to support that with pictures like this, where you really see that the RT deviation as a function of the gradient length. As you go along the gradient, the RT deviation is not really linear. You often see things like that going on. And thus they claim you need to do nonlinear alignment. And that is, to some degree, that is true. But I would like to point out that we don't need to bring them exactly on top of each other for the linking. We just need to bring them close enough together that in this local neighborhood, there are not that many matches. And with high resolution data, the odds of finding a feature that has exactly the same mass just needs to be small enough. Yeah? So you don't need to get the error in the retention time to 1 second or less. You just need to bring it down such that the map is sparse enough that you can identify these guys properly. So we see some difference in linear versus nonlinear, but for most applications, it makes no difference whether you use linear or nonlinear. What you see is that the retention time error goes down, but that is to be expected. Yeah? But you can do things like lowest regression, you have a much reduced RT error, but actually for the linking of peptides across maps, the effect is much less pronounced. The feature linking can be done in various ways. For label-free quantification, you do across maps, but you can also imagine for labeled quantification, you can also link within the map when you're looking for pairs, Silac pairs or Silac triplets. It's the same idea, same algorithm underneath it. Basically, what you need to know is some sort of mass tolerance. So what's the resolution of your instrument? And then you need to know some retention time tolerance, and it's just going to pick up these pairs and put them all together into one consensus map. So what we're doing in OpenMS is we are implementing this post-clustering algorithms. We also have others, nonlinears, that we will not look at here. There is a map aligner post-clustering, and there's a feature linker unlabeled QT that is being applied, and we will use that in the pipelines and workflows for quantification. What comes out are so-called consensus features, and this is what you see then in top view. You see these lines. When you zoom in, you will notice that these lines are more stars. They look like lines because the variation in retention time is, of course, much higher than the variation in M over Z. So when you zoom in, you see, of course, M over Z scatters as well. But the scatter is much lower than the retention time scatter. But you can see this here is a so-called consensus map. These dots of this star here, these are features in different maps. And the center here, that is sort of the consensus feature. This is where the center of this particular feature is across all maps. And these are all the features that were pulled together to be one thing that can then be quantified across all of these maps. If you do quantification, then one thing you really need to do when you go beyond, say, 10 maps is quality control. You will have, if you measure 1,000 samples, you will have samples where something went wrong. And if you try to align these guys with all the rest, then it's not going to be helpful. I really recommend doing some quality control. We also have quality control in this tutorial. I will not talk about it right here, but I can only encourage you to look at that. It's really essential. If you don't do it, you run into trouble. You can do that with various ways. You can do simple map statistics. And there are a few things that I would like you to sort of keep an eye on. One thing is you can look, just by looking at this consensus map, you can extract some statistics. And these are distributions. This is the distribution of the mass to charge shift. That's basically the stability of the mass within the same feature. And if you see that this goes up beyond what you would expect from your instrument accuracy, then you have a calibration problem. And in this case here, we see this is the mean is certainly below, probably below 0.5 ppm, so that looks fine. But if you see that going all over the place, then you got an issue. This here is a distribution of the delta RTs. So by how much are these different features away from the center? What would you expect here? Well, you see this is, for most of these maps, this is below 10 seconds. So for regular long enough gradient, that is still fine. But if you see that this going up to minutes, then again you might have a problem in your chromatography and the reliability of the chromatography. That can easily happen if someone switched the columns within the experiment. And then it might be good to correct for that by aligning the first batch and then aligning the second batch independently, and then think about how to bring them together. Another thing that I would like to point out is you will not always see everything in every map. And that is a bit disappointing to many people, this particular plot. We call it the Bathtub Plot for the obvious shape. So what's plotted here is the number of features on the y-axis and the number of maps in which these features have been observed on the x-axis. So what would this plot look like in an ideal world? In an ideal world, there would be only one bar right here. So we have 20 maps, and everything occurs in every map. It doesn't look like that. So what's going on? I mean, this looks scary to many people, but it's really not. What's going on here? Shouldn't you expect that sort of the peaks are highest here on the left-hand side? So who would have thought that the highest peak here in this distribution would be at 1? What does that mean? What does 1 mean? Well, 1 means the vast majority of features is found in a single map and a single map only. It's found nowhere else. These are low intensity features that are close to the level of detection for the feature finding. And they basically pop in and pop out. Now you can change the parameters. You can make your feature finding less sensitive. Then they will they disappear? There will be fewer. Yeah? They will not disappear. And you can argue at length, are these guys false positives? When you look at them, you will see, no, there is signal there. And are they what you're interested in? Probably not. Because if you see them only in one map, then you cannot really quantify them. So what you usually do in downstream statistical analysis, you need to do some sort of stratification and really get rid of them. But what you will never see if your sample size is large enough that you see a feature in all samples. It won't happen. You need to be able to deal with missing values, because no matter how hard you try, some of these peptides will not be present in some of these samples. Okay. That brings us to another issue that we can only touch on briefly, but since there's so much statistics going on at the May Institute, I thought I would stay out of that. I would just give you some pointers. And again, we are happy to discuss it later on, and also Olga can help a lot with that. We will process some of the OpenMS data also in R, in MS Stats, in other parts of the May Institute. There are different ways to deal with normalization, and that's a big issue in label-free, because you have no internal normalization as you have in SciLec or iTrack. There are different ways how people have addressed that. One is spike in peptides or proteins and make sure they are on all maps and then normalize on that. Doesn't really work all that well. You get tons of other issues. It's much easier to do what we like to call background normalization. And the reason is homeostasis. We talked about that earlier. It's actually very unlikely that the majority of the proteome shifts. Housekeeping proteins are essential. And if you don't have a tissue that is completely dead anyhow, which is not a typical application, then you would expect that a large chunk of these proteins will be the same across all your samples. And you can use that for normalization with the background. And you shouldn't be scared of, say, even half of the features change. That does not matter. This background will still be able to sort of pull it all back. The trick is here to do some sort of robust regression. You basically look at all the ratios. If you have two maps, for simplicity's sake, you look at all the ratios in these maps. and you get some sort of distribution. Some of them will be upregulated, others will be downregulated, but the majority will be right in the middle. And with a robust regression, we mean we discard everything that is regulated, and we look at the ratios again. And this way, we keep only the proteins that are really in the middle, that are preserved at a more or less fixed ratio, that converges in a few iterations, and you keep those guys to really normalize all the maps. And the ones that are up or down regulated, you don't consider them for the normalization. But it gives you a much broader basis than having just 10 peptides that you spiked in with all their issues. It gives you 10,000 proteins that you can normalize with. And that is usually a much more robust way of dealing with that. And then, of course, you can do other types of normalization. There are other issues like fractionation that we can't go into. But this here is an example where we did that. That's out of the platelet paper I mentioned a while yesterday, where you see that if we have here stock concentration and intensity, before normalization we do have some scatter, and after normalization you see that the concentrations look really neat and are more or less on a straight line. And that was just done by background normalization on all these other proteins. If we implement these workflows in KNIME, then that is usually a straightforward workflow. What you need to do is use all the features, all the runs, all the MZML files, have a loop, apply the feature finder tool, apply the aligner tool, apply the feature linker tool, and then you write out the map. That's it. It's that straightforward. But with label-free quantification, that's where the fun only starts. Then you start doing normalization, you start looking at the proteins, you start looking at batch effects and so on. But that is downstream of the feature finding. All right, I'll stop here, and I'd be happy to take any questions. Yes?
How many samples you usually work for this like metabolomics or like in order to get a meaningful like for label free, like for some annual research or annual free or like for cancer research? And how many samples are there?
How many samples? I mean, as many as you want. It can be 2, but if you want to get statistical power out of that, you usually need more.
5 at the starting.
5 is a good starting point, but you don't need 5 biological replicates. It really depends on the question that you're asking. And we can talk about replication and how to do that offline, I think, because that's a topic on itself. But label-free is typically applied to larger sample sizes, 20, 100, 1,000 samples. That's what we see and what we can easily process.